Hello, my name is Miguel and I'm going to talk about the PCR. PCR means polymerase chain reaction. It was developed in 1983 by American biochemist Kary Mullis. He was awarded the Nobel Prize in Chemistry in 1993. PCR is using molecular biology to make many copies of small sections of DNA or gene. There are three main stages. The first one is denaturing. It's when the double-stranded template DNA is heated to separate it into two single strands. The second one is annealing, when the temperature is lowered to enable the DNA primers to attach to the template DNA. The last one is extending, when the temperature is raised and the new strand of DNA is made by the TAQ polymerase enzyme. A complete PCR reaction can be performed in a few hours, or even less than an hour with certain high-speed machines. PCR has many research and practical applications. It is routinely used in DNA cloning, medical diagnostics, and forensic analysis of DNA. Some advantages are: it is valuable for detecting specific pathogens that are difficult to culture in vitro. It is significantly more rapid in providing results compared to culturing, and it's a valuable screening tool. Some limitations or disadvantages are: PCR testing alone may be limited as a diagnostic tool. The second one is post-treatment diagnosis may be challenging. And the last one, false positives and false negative results. Thank you.
Hey, I'm Mariano and I will talk about the cloning process and give you some examples of it. Well, I can start telling you that researchers remove a mature somatic cell, such as the skin cell of an animal that they wish to copy. For example, a dog. They will transfer the DNA of the donor animal somatic cell into an egg cell that has its own DNA containing nucleus removed. Researchers can add the DNA from the somatic cell to an empty egg in two different ways. In the first one, they use a needle and they inject it into the empty egg. In the second way, they use an electrical current to fuse the entire somatic cell into the empty egg. In both processes, the egg is allowed to develop into an early stage embryo in the test tube, and then is implanted into the womb of an adult female animal. Finally, the adult female gives birth to an animal that has its same genetic makeup as the animal that donated the somatic cell. In other words, the male. This young animal is referred as the clone, of course, and reproductive cloning may require the use of a surrogate mother to allow development of the clone embryo, as was the case for the most common clone organism, Dolly the sheep. But I know a few examples of a successful cloning besides Dolly. For example, I found a family of pigs. I found it very interesting because it was a few organisms. They were 5, if I'm not wrong. Millie, Alexis, Krista, Dotcom, and Carol. Yes, 5. Then I found a mouse named Cumulina, Nodo and Kaga. They were cows. And Tetra, the rhesus monkey. This is very interesting. Well, I'll let you continue with my partner Byron. See you later.
Hi, my name is Byron Susek Mombela de la Garza, and I'm going to talk about positive and negative aspects of cloning. Well, in the positive aspects, we have that infertile people or same-sex couples could have children made from cloned cells. Another thing is that a clone could be a source for transplant organs or tissue. Cell cloning could assist scientists in gene editing and research. Other thing is that after editing or removing bad genes, cloning could lead engineering humans for specific traits. Other thing is that cloning could enhance and advance human development. I think there's a lot of more positive aspects, but these are the more important or the more general that I found. In the negative aspects, we can find that the negatives of human cloning, including the making of designer babies, and then we have that could be a violation of the clone's individual human rights. Another thing is that cellular degradation occurs when too many clones are made from embryos. Other thing is that cloning raises the question of a moral or human right to an exclusive identity. Another thing is that human cloning could produce psychological distress for the clone and society. I think cloning is the future, but we have to fix it to be all good things. I'm going to talk about the prenatal genetic screen. It is the test that the pregnant women use to see their babies in her stomach. This is also known as ultrasound. The use it normally is used in the blood testing, a check swab, and amniocentesis. This is only for pregnant women that are involved to the use of a thin noodle inserted into the uterus to collect all the fluids. This is only for the pregnant women. We have the pros. These pros are like the more significant ones. We can see the baby after he or she is born. We can see if it is boy or a girl, and this rise in the popularity, and it is easy to understand. It is a file that offers numerous benefits to the doctors and patients. This is too easy, as we know, and we also have the cones. These one are like, well, it is not that dangerous, but it can also be a possibility, right? So it could be some risky to, if we see it, these have the ethical concerns held. The concerns and legal concerns have surfaced with regards to the genetic test. This prenatal genetic screen has more benefits than cons.